So this webinar comprises 2 sessions. The first one is today, which will last from 8:30 to 10:30 in the morning. And the next session will be next Wednesday, 10th of November, from 1:30 to 3:30 in the afternoon, everything UK time. As well, you remember that if you haven't registered for the next session, you have to do so if you want to attend. Today, we will hear 4 world-leading experts in the field of sleep and mental health. First, we will start with Professor Sandra Kooij, who will present her work on sleep and ADHD. Then this will be followed by Professor Sean Drummond, that he will present his recent work on the mechanistic role of sleep in post-traumatic stress disorder. Then Professor Tina Paunio will talk about the genetic background for sleep and psychiatric problems. And finally, we will end this session of today with Professor Timo Partonen, that will present some of his work on chronotype and mental health. Each of these talks by the speakers will last 20 minutes, and after each of their presentations, we will have 10 minutes for Q&A. So please, during the talk of each of the speakers, you can leave your questions in the Q&A section that you can see at the bottom of the screen, of the Zoom screen. And then at the end of the speaker's talk, we will go through the Q&A section as much as we can in these 10 minutes. And then if you have some doubt that has not been answered, then you can raise your hand on Zoom, I think, and then we can allow you to ask a question if there is time for that. And now I will introduce the first speaker of today. So it's a real pleasure for me to introduce all of you, Professor Sandra Kooij. Thank you very much, Sandra, for attending or for accepting this invitation for the webinar. Sandra Kooij is Professor of Psychiatry at the Free University in Amsterdam, and she's also the founder and chair of the European Network Adult ADHD and chair of the section of neurodevelopmental disorders at the European Psychiatric Association. She is one of the pioneers of investigating sleep problems in ADHD in adults, and today she will talk about how sleep, ADHD, and health are closely interconnected, and the necessity to educate, treat, and prevent ADHD and sleep disorders. So now, Sandra, this is this is your time, and you have 20 minutes, and we're looking forward to your presentation. Thank you very much.
Thank you very much for the invitation. I'll share my slides with you. Do you see them? Okay, wonderful. I will talk to you about ADHD, sleep, and health because they're closely connected. I have nothing to declare. Yes, my first quote is, everything is timed, timing is everything. And living out of phase, that means being a late or early bird, means jet lag. And jet lag is not only something that we experience when we fly from the United States back home, but it's something our body experiences every night that we sleep late or out of phase compared to the rhythm of the world around us. So this picture shows you just which processes are timed processes in our body. So the temperature is lowest at 4:30 in the morning. Our blood pressure rises at 6:45. Our melatonin secretion, the sleeping hormone, stops at 7:30 when light enters the eye. Our bowel movements will start up after breakfast. And so everything is nicely timed. Coordination, blood pressure, bowels, and so on. Reaction time, concentration issues, everything is timed. So if we make a mess of our sleep pattern by behavioral changes, we are also jet lagged. And jet lag, we know, is very unhealthy. So this is the first start to wake you up. For those of you who are not familiar with ADHD, just a short summary. ADHD starts per definition in childhood, has a chronic persisting course of mainly 3 core symptoms: inattention or forgetfulness, chaos, hyperactivity, restlessness, and impulsivity. But there are more symptoms around that are also highly frequent, like binge eating, mood instability, and mind wandering. It's a horrible condition in most cases, inducing a lot of impairment lifetime, a lot of psychiatric comorbidity, and a lot of associated physical diseases. Now about sleep. Should I minimize this? Yeah, I think so. Now about sleep. When you look into the literature, there has been done a lot of research already in children with ADHD that have sleep problems from childhood, from early childhood on. And the most frequent disorder that you find is the extreme evening chronotype or delayed sleep phase disorder. We measured this by measuring melatonin in saliva, the melatonin onset, and we found this disorder in 78% of a consecutive sample of adults with ADHD. I'll show you the data later. In children, a similar number was found, also in the Netherlands. Insomnia, which is difficulty falling asleep by ruminating, worrying, thinking about everything you have to do the next day, is also highly prevalent, 43%. Obstructive sleep apnea, 30%, restless legs, 30 to 40%, narcolepsy, enuresis, bruxism, nightmares, everything is increased. So this induced in my head the question, is ADHD in fact a sleep disorder? Why didn't we see this earlier? It could be that ADHD causes sleep problems because ADHD means restlessness, a restless brain, restless thinking, rumination, worrying all the time. So maybe that is the reason that you can't fall asleep, or the sleep problems cause ADHD symptoms. It's also possible because if you have sleep debt due to short sleep duration, you will have cognitive and mood issues that are very similar to ADHD. Or are they both interconnected? And is there a reciprocal causation? I tend to think that at least this is true, but maybe it's even true that ADHD and sleep problems share an underlying etiology. And we are trying to find out what's the real truth about ADHD and sleep. So here's a short summary. 70 to 80% of adults has one or more sleep problems. 78% sleeps too late and cannot sleep at an earlier time. This is related to a 1.5 hour delayed onset of melatonin in saliva in ADHD children and adults. It starts in childhood. This sleep pattern also, it's heritable. It's called chronotype. You will hear more about it during this webinar. There's a social jet lag based on the fact that we have to get up early in the morning for school or work or other obligations. And this leads to a sleep debt, because if you sleep late, let's say at 2 or 3 in the morning, and you have to get up early, you only sleep maximum 5 to 6 hours on a chronic basis. And we all know optimal sleep duration is 7-8 hours to stay healthy. Because the consequences of short sleep are ADHD-like symptoms: sleepiness, less focus, lower cognition, impaired memory function, irritable, sad mood, and binge eating is a direct effect of sleep loss every night. So in fact, sleep debt and ADHD overlap. These are the short-term consequences of sleep debt, but in the long term, you get fat, you grow fat because of sleep loss. Because if you are not, if your brain is not cured by rest, by sleep, it demands food for energy, I think. And this has been shown by internal medicine, showing that the leptin and ghrelin levels are out of order, leading to increased appetite. Same is true for winter depression. But obesity is the porte d'entrée for diabetes, hypertension, cardiovascular disease, cancer, and Alzheimer's. So all chronic diseases that you want to avoid are starting with obesity. Not only, but it's a main porte d'entrée. So we should stay out of this disease. Having said that, all sleep disorders are more prevalent in ADHD. We should, and all sleep disorders lead to sleep debt that has a common ground for all those physical diseases. We should remember that every disorder, every sleep disorder has a specified treatment that's proven effective. So we should not only do sleep hygiene for people with sleep problems, because in delayed sleep, we give chronotherapy. We reset the clock. I'll talk to you later about it. In insomnia, we give CBTI, cognitive behavior therapy for insomnia, but it's proven effective for this group. But most people with ADHD will have this combination, so they need both. Restless legs in 30% needs ferritin suppletion if there's a low level, and or pregabalin, which is very effective in 1 week. Sleep apnea during sleep requires weight loss, mandibular device, or continuous positive airway pressure. We developed this sleep protocol in our psychiatric department in order to teach ourselves, our psychiatrists, how to treat sleep problems. And most of the sleep problems we can treat ourselves. The only sleep disorders that we cannot assess are sleep apnea and periodic limb movement disorder during the sleep time. But the rest of it we can treat ourselves. So this helps patients to get an integrated treatment and not having to wait for ages for a sleep clinic assessment and treatment. Now, about the sleep wake disorder, the late sleep disorder, the circadian rhythm sleep wake disorder, as it's mentioned in the DSM-5, is characterized by being a late evening type, a late chronotype, having a chronic pattern of late sleep and late rise, leading to daytime sleepiness and or difficulty falling asleep on time. People compensate by sleeping early, late, in between, and again. So they mess up the rhythm completely by compensating for sleepiness. And this leads to dysfunctioning due to increased inattentiveness and or social problems. Well, what about the circadian rhythm? Most of us are not knowledgeable in this area. Well, the rhythm is set by light, and the light enters the brain through the eye and reaches here the ganglion cells in the retina that we call the melanopsin ganglion cells. Melanopsin is a compound that measures light intensity. So the light intensity is measured, and through the chiasma the supranuclear chiasma opticum, the light intensity is communicated to the pineal gland and to the clock, the biological clock. So we in fact know by the intensity of the light that we perceive what time it is. The brain knows it. And the brain starts all those processes that are needed to have no jet lag and be synchronized with the time of the world. The sunlight, the light every day induces energy, better mood, and better sleep at night, because we need contrast between day and night. We need sun during daytime, and we need darkness, complete darkness at nighttime. So the chronotype is your genetic makeup for being a late or early person. And usually in the population, it's no more than 20% that is late. And in ADHD, it's 80%. So that's a huge difference. The time of the light onset in winter or summer is different, and this induces a later or earlier onset of waking up. When the light enters the eyes, melatonin stops, melatonin production stops, and dopamine, that's the compound that's important in ADHD, that is our daytime hormone or neurotransmitter, it wakes us up. And this starts when melatonin stops and the light is on. In the evening, the reverse occurs. Melatonin starts and dopamine is set off. And this circle is occurring every 24 hours in our day and night cycle. The eye, it's interesting to know, not only measures through melanopsin light intensity, but also the eye contains melatonin and dopamine receptors. Both communicate with the clock, telling the clock what time it is. Isn't that interesting? This is our first study from 2010. We measured the dim light melatonin onset in saliva in a consecutive sample of 40 adults with ADHD. And we asked them whether they had sleep onset insomnia problems, and we compared it with healthy control data. And we found ADHD only had their onset at 11:15, healthy controls at 9:30. This is the mean in our country for the onset of melatonin. And it means that after 2 hours, you want to go asleep, to fall asleep. The ADD type had more long sleep and less difficulty falling asleep. And their onset of melatonin was also delayed, but less delayed than the ADHD type. So the subtypes of ADHD were separated by the sleep pattern, by the chronotype. I think that's also very interesting and new. We not only measured melatonin, we also measured movement patterns using actigraphy. And here you can see the moments of sleep. There are the blue lines where people lay still because they are asleep. And here you see daytime movements. We were able to show that the ADHD people with sleep onset insomnia had a shifted movement pattern to the right of 1 hour and a half. So not only melatonin was 1 hour and a half delayed, also the movement pattern. And what else is late? Well, appetite is late. People have no breakfast. ADHD people skip breakfast often as a rule because their stomach feels asleep. The temperature profile we measured in the next study has the same picture, is delayed 1 hour and a half. So this is the movement pattern and this is the delayed melatonin. Could it be that ADHD symptoms are the diurnal and late sleep is the nocturnal presentation of the same problem? So is it a sleep disorder or more specifically a disorder of the clock? And could we improve ADHD by treatment of sleep problems? Interesting. Well, here is an indication for that. This is a study by Martijn Arns. He showed this is the United States. The prevalence of ADHD is higher here in the East than here in Mexico. And he put this map of the light intensity of the same country. And here it shows that the light intensity is highest in Mexico. And what has this to do with ADHD? Well, the idea is that light intensity leads to better sleep or consolidated sleep pattern and rhythm, leading to less ADHD severity. And that means that the prevalence of ADHD is indeed lower in this area. And indeed, he found that 34 to 57% of the variance of ADHD prevalence could be explained by solar intensity. So this is an idea about how to consider the interaction between sleep loss, sleep duration, and sunlight, and ADHD severity. Well, what's more important also is that late sleep and health are connected because of this associated appetite increase and the obesity and other disorders. And this is, I think, what may be the cascade of events. If you are ADHD person with obesity, you have 30% chance of seasonal affective disorder that's associated with carbohydrate craving. And you have late sleep in 80%, leading to short duration of sleep, altered leptin-ghrelin ratios, leading to carbohydrate craving, breakfast skipping, binge eating. And I think the factor behind all these disorders might be inflammation, as we currently think that is the case in obesity. We assessed physical disorders and complaints in our patients a few years ago, and we found that they had significantly more problems on all areas that you can think of. And this was recently confirmed in a Swedish registry study by Durit, and they showed that on all systems, ADHD people had a 2 to 3-fold increased risk. And they, in fact, had 34 of 35 physical conditions more frequently than controls. Now I'll tell you something more scary. Melatonin is very important, not only for falling asleep, but it's also an antioxidant. It's related to the metabolic system, so maybe also to appetite. And it inhibits at night cancer cells to grow and to develop further in our bodies. And shorter sleep duration, as we have seen in our patients, may mean that they have a shorter exposure to melatonin, to protection actually. And then in 2015, I read this paper, Melatonin, an inhibitor of breast cancer. And I thought, how is it possible that nobody talks about it? But now we see many, many more papers on melatonin the treatment of cancer. And it's not instead of chemotherapy or surgery, but it's an added treatment that seems to be effective. So it's something to consider before you think, I mess up my rhythm, I go to bed late, I have short sleep. Doesn't matter, it does matter. Because we want to prevent negative health outcomes. So I'm out of time, I think, but I'll go on a bit faster. So how do we treat this late sleep? Sleep hygiene, melatonin, and light. Sleep hygiene is maybe easier to understand for you. Let your day be bright and your night as dark as possible would be my general advice, meaning that we mimic Mexico. So contrast is important. Make your bedroom as dark as possible or use eye masks. Block all light sources, blinded curtains, no chargers, timers, or mobile phones that emit any light that might wake you up, because we are very sensitive to light, even through closed eyelids. Use no screens after 9:30, the time that your melatonin should increase. Or if you do need to use TV or screens, use orange goggles. Here they are. Sorry. To prevent the blue light of the screens enter your brain. And this, my patients tell me that it's effective if you start early in the evening at 7 p.m. You can use the screens, but you should continuously do that and not forget it. Well, there are some more tips for temperature control: hot shower at night, not in the morning. Try to wake up every day at the same time. Aim for 7-8 hours between 11 and 7. No napping during daytime longer than 30 minutes, because it reduces sleep pressure that you also need to fall asleep. Go outside in the early morning, walk the dog, and so on, and limit use of sunglasses during daytime. My patients are oversensitive to light and wear sunglasses in 70% in every season, more hours a day than controls. So there is something in the eye going on as well in ADHD, and this is our next study. Melatonin, we use 1 to 3 milligrams maximum. It's a high dose at 10 p.m. using a timer every day on the same time, because it works on the clock. Don't use it after 12 at midnight, because it may reverse the rhythm. It only works 3 to 4 hours. It's a high dose. And when you're sleepy in the next day, the dose is too high, you should reduce it. For some people, they still wake up after falling asleep with melatonin at 3 a.m. and they could benefit from long-acting melatonin, like Circadian in our country, 2 milligrams. Light therapy is typically used in the morning, early morning, at a fixed time, let's say between 7 and 8 a.m., not later, because it works on the clock. It must be white or blue light, at least 10,000 lux, and it should be close to the eyes if you use such a light box, because the distance is essential, and the duration, 30 minutes, and the time for the effect. You use light for 1 to 2 weeks for winter depression, late sleep 3 weeks. And when you stop the light, the rhythm returns to its previous level. So you will be late again in 2 or 3 weeks. We just studied that. These are good lamps. We use these glasses nowadays because it spares time. You don't need to sit still in the early morning when you need to go out for work and have your children getting ready for school. The wake up is typically not effective because it has a too low light intensity. This is the website of the glasses we use, and we are currently studying them. Chronotherapy indeed does reduce ADHD severity, but not to the same extent as stimulants do, because nothing can beat stimulant effects. But we published this paper on chronotherapy and the effect on the severity of ADHD. More information in my newly updated book, Adult ADHD, some webinars on YouTube, and well, have fun. Thank you very much.
Thank you, Sandra. Thank you very much for such an exciting presentation. And Matthew, now maybe there are, yes, I think maybe 5 minutes for some Q&A if there are some. So now I leave it to you. Thank you.
Yeah, thank you very much, Professor Kooij, for a fantastic talk. I really enjoyed it and learned a lot from it. So thank you very much indeed.
You're welcome.
I will just check the Q&A. So nothing as yet, but a couple of questions I was thinking of as you were talking, Professor Kooij. I mean, one thing as a clinician is how responsive are the sleep disturbances you mentioned to the sort of traditional treatments of methylphenidate and stimulants? I guess that's the one question. The second was, I've been visiting universities in England with my son for open days and just seeing students sitting around with cans of stimulant drinks next to their laptops and study areas. I just wondered, again, how much of the sleep disturbance is linked to self-treatment through, as I say, non-prescribed stimulants. So I'll be grateful for your thoughts on those two items, if that would be okay.
Okay. Well, first of all, the stimulants do have an effect on the timing of sleep. That has been proven in fibroblasts by Andrew Kooij from your country, but not only by him. But it's a relatively little effect. It's a small effect. So the pre-existing disorder of the late sleep chronotype has a huge effect. It's chronic, it's lifetime, it is detrimental. So the effect of the stimulants is only a small effect compared to this pre-existing sleep disorder. So take it seriously and don't blame the stimulants for something that has been there all their lives. Now that I tend to think of ADHD as a day and night problem, as a 24-hour problem, as a sleep problem, I treat ADHD with stimulants during daytime that have to wear off at night, and then give melatonin. So then we have a more on-off phenomenon induced by this treatment for ADHD and the late sleep, the chronotypes problem. So then we get a fixed rhythm in the morning, waking you up with dopamine, methylphenidate or dexamphetamine, and getting you asleep on time with melatonin. It may seem a bit artificial, but it's very helpful, I can tell you. And it produces most stabilized patients I've ever seen. So then about the students that use the stimulants for cognitive enhancement. That's a nice term that we now have learned that it exists. It happens, and methylphenidate is sold on the black market. Students that want to study during nighttime to get the best results for the exams. Well, I have to disappoint them. Studying at night is not so well for your long-term memory, and you will forget about it after the exam immediately because your memory is not stored well enough. And so it's not a good investment, in fact. But we all want to try, of course, to do as little as possible to yet get our exams done. And we cannot prevent it. And I think stimulants are not as dangerous as people think. They may be dangerous for people with underlying conditions, heart conditions, but most young people don't have that. So I think most students that do use the stimulants may be students with ADHD that are not diagnosed. And they can easily get the medication from their brothers and sisters that have the disorder. So that makes sense that they can easily have access to the medication and then that they need it more than others. So I think the advice would be, if you profit from stimulant medication, get yourself a proper diagnosis and good treatment.
Thank you, Professor Kooij. I'm aware we're close to time, but a couple of questions coming from attendees. So maybe I'll put those to you if that's okay, and
Sure.
where you need to head off to clinic shortly. So a question, maybe I'll package them together. So a question from Rory. Thank you for the brilliant talk. Thinking back to the idea that ADHD behaviours and sleep difficulties may have a common underlying cause as opposed to causing each other, is there any evidence linking melatonin release patterns to daytime ADHD behaviours? So that's Rory's point. And Michaela has also got a question. Do you think ADHD reward-seeking behaviour in the evening contributes to sleep problems, as this is the reverse of what the body is trying to do through attempting to restrict the release of dopamine? So I guess, is it, I guess when you have ADHD, you're trying to keep yourself stimulated at night?
Yeah, well, I think due to the ADHD chronotype, you are more active at night and you're sleepy in the morning. because your clock is delayed. So it's normal for ADHD to be very active at night and thereby also having behavior that doesn't help to fall asleep. And adding to that the screen use that you do often, because it's dark, you do use light and screens. So it's adding up. And it's not only behavior, there's genetic background for this sleep pattern, and it's lifetime. It's highly chronic. So if you want to change it, you really have to be convinced of the need to do so. And I tend to think we should do that to prevent all those chronic diseases that may result from it, including severity of ADHD that's increased by sleep loss. And the first question was, sorry.
I'm sorry, that's my fault, Professor Kooij. So is there any evidence, this is a question from Rory, linking melatonin release to daytime ADHD behaviors?
Well, we know that a melatonin release is late in ADHD and that it's more prevalent in ADHD. So it's about 80% that has a late onset of melatonin, whereas it's only maximum 20% in the population. So yes, there is a link.
Mm-hmm. Thank you. I'm aware we've just gone past 9 UK time, so I think we better pause there. We had a question from Odessa. I'm sorry, Odessa, we couldn't get to you, but a massive thank you to Professor Kooij for a fantastic talk and start to our webinar today. So thank you very much, Sandra, for your time and your input.
Thank you so much. Have a great day. And I will look back your presentations because I'm not able to attend for now. Thank you very much. Bye-bye.
Thank you, Sandra. Bye. So now it's done of Professor Sean Drummond. So now Professor Andy Baxo will be the person introducing Sean. Thank you very much, Andy.
Thanks, Isabel. So Sean joins us from Monash University of Melbourne. Many thanks for joining us this late because it is 8 p.m. and then he should be settling down to bed. He's Professor of Clinical Neuroscience and Director of the Sleep and Circadian Rhythms Research Team within the Turner Institute at Monash. He's got wide-ranging interests in sleep, from cognitive neuroscience of sleep and sleep deprivation to treatment of insomnia. And today he'll be talking to us about some of the work he's been doing, looking at the mechanistic role that sleep plays in PTSD. Thank you, Sean.
Thank you very much, Andy, and thank you, Isabel, for the invitation to present today. Just so we're all good. We can see in here. Excellent. Thank you, Andy. Okay, so as Andy said, I'm going to talk a bit about some of the work our group has done to try to better understand the connections between sleep and some of the fundamental fear processes that underlie not just PTSD, but all of the fear-based anxiety disorders as well. And these fear-based processes, abnormalities in these fear-based processes are a hallmark of PTSD. So as folks may know, there is increased quantity and generalization of fear conditioning in PTSD. So people will have a fear response to lots of stimuli in their environment. At the same time, there's impaired fear inhibition. And fear inhibition sort of takes two flavors. One is fear extinction, which is the ability to learn that a stimulus that you currently fear in your environment is no longer a threat and should not elicit a fear response. That's fear extinction. Safety signal learning is kind of the mirror of fear, whereas a safety signal is a stimulus in the environment that signals the active absence of danger or the presence of safety. And the ability to learn both extinction and safety are impaired in PTSD. And there's a rich animal literature that has looked at the relationship between sleep and these fear processes. And what they show is if you fear condition an animal, typically it's a rodent, but not always, when you fear condition an animal, it will fragment and shorten REM sleep in the very next sleep episode. At the same time, if you fragment or shorten or deprive an animal of REM sleep, it disrupts the ability to learn extinction or to unlearn fear. And so you get this positive feedback loop where an animal is fear conditioned, it disrupts REM sleep. That very disruption in REM sleep then impairs the ability to engage in extinction, which allows the fear to dissipate. And there have been growing number of studies in humans now that have basically also shown a relationship between REM sleep and either the ability to learn extinction or the ability to remember extinction the next day after you learn it. When we started the work that I'm going to show you today, there were no studies in any species that had looked at the relationship between sleep and safety signal, which is the other side of fear inhibition. And there was, and to some extent still is, this controversial and I would say somewhat misguided hypothesis that because sleep plays a role in memory consolidation, if we sleep deprive somebody after they're exposed to a trauma, that will impair the consolidation of the trauma and therefore prevent the PTSD. And so in the papers that I'm going to show you now, we wanted to address all three of these questions. Now, before I jump into the sleep-related papers, I want to make sure that everyone understands how we measure fear conditioning and extinction and safety. And we use something called a fear-potentiated startle paradigm, which is a really well worked out and well-used experimental paradigm in the literature. And when we do it, it's 3 days. So on day one is the learning of fear and safety, or the acquisition of fear and safety. And what happens is participants see a series of blue and yellow circles and blank screens. 75% of the time that they see a blue circle, we shock them on the wrist or provide some other aversive stimulus. And so after, a handful of blue circle shock, blue circle shock, blue circle shock, the person figures out that, hey, when I see this blue circle, I'm about to get shocked. And so the blue circle itself will elicit a startle response or a fear response. Within the same task, the person sees the yellow circle a bunch and they never get shocked. And so after enough pairings, the individual learns yellow is signaling the absence of danger or the presence of safety. And so they actually start to show a safety or a relaxation response when they see the yellow circle. And then the blank screens really just serve as a baseline for these other two. So day one, they're learning the fear, they're learning the safety. Day two, they come back, they see all the same stimuli, but we never shock them. And so day two allows us to do two different things. First is we can look at the extent of overnight memory consolidation for the fear and the safety signal. Do they remember those today? So the first few times they see the blue circle, if they remember that it was a threat signal yesterday, it meant they were going to get shocked, they will show a fear response on day two. At the same time, if they remember that the yellow circle, meant safety yesterday, then the first few times they see the safety signal, they're going to show a kind of a relaxation or safety response. So we can look at overnight retention. And then as we continue to show the blue circle with no shock on the wrist, they will eventually learn, oh, wait, the blue circle no longer is a threat signal. I don't have to show a fear response to this. And they will start to instead show a response very similar to the yellow circle. And that is the process of developing an extinction memory. Then they come back again on day three. Day three are really only concerned about the blue circle, but again, they see everything, they never get a shock. And the question is, do they remember that the blue circle is no longer a threat signal? In other words, do they have intact extinction recall? If they do, they should show a relatively small fear response on day three. If they aren't able to retain the extinction learning, then the fear will come back and they'll show a big fear response on day three. So this is the basic paradigm that we use in the studies that I'm going to talk about today. And the protocol that led to the data that I'm going to show you is this multi-day protocol. So individuals, these are all healthy controls. They lived in the lab for 4 nights and 4 days. The first night and day, not much was going on. It was adaptation. Then they had a baseline night of sleep. Day one, we did the fear and safety acquisition. So they learned both the threat and the safety signal. And then they were randomized to one of 3 groups: a normal sleep group that slept normally throughout the protocol, a pre-extinction sleep deprivation group. So immediately after fear and safety learning, we sleep deprived them for 36 hours and then tested both recall of the fear and extinction learning. Then they got to sleep and we did extinction recall. And then the third group is the post-extinction deprivation group. Here, after the fear and safety learning, they slept, they did extinction learning. After the extinction learning, then we sleep deprived them for 36 hours and tested the effect on extinction recall. Now, the first set of analyses that I'm going to show you, we were only really concerned about the first 2 days and only concerned about the groups that slept every night so far. So we collapsed these 2 groups together because effectively they've been treated exactly the same so far in the protocol. And so as a reminder, they had an adaptation day and night. And then on the baseline night, we were particularly interested in measuring REM sleep because of the data coming out of the animal literature. We did the fear and safety learning on day 1. Then we looked at the change in REM sleep on the next night relative to baseline because we wanted to see if we could replicate this animal data that says if you fear condition an animal, you disrupt REM sleep. Then we looked at day 2 to see whether the REM sleep at night played a role in overnight memory consolidation for the fear or safety signal. In the 2 arms of that study, we had an N of 42 young adults, reasonable proportion of women and racial ethnic minorities. And then for the analyses, we were interested in a number of both explanatory and response variables. So from the fear-potentiated startle task, we were interested both in the ability to learn fear and the ability to learn safety. And then on the REM side, we were interested in 3 different REM variables that, again, came out of the animal literature. And so because we were interested in a bunch of variables, we used a canonical correlation because a canonical correlation asks, is this group of variables A correlated with this group of variables B? And if they are, which members of A and which members of B are driving that relationship? So schematically, it looks like this. On day 1, we were interested in both fear learning and safety learning. The analysis creates a latent variable out of these 2 called startle acquisition. Then we looked at sleep that night. We had 3 different REM variables. They were turned into a latent variable called REM sleep. And the first question is startle related to REM that night? The answer was yes. We had a pretty robust correlation. And so then the question is, which of these variables are driving that relationship? And to our surprise, on the startle side, the fear learning was not at all related to REM sleep that night. Instead, the relationship was driven entirely by safety signal learning. Then on the REM sleep side, all 3 variables here were related, as it turns out, to the safety signal learning. And so in essence, what we saw is that stronger safety signal learning during the day led to greater REM consolidation that night. And the reason we call it REM consolidation is it was increased REM efficiency, increased REM percent, and longer REM latency. And so safety signal learning accounted for 28% of the variance in REM sleep that night, which is pretty robust for this kind of behavioral study. But it still begs the question, so what? So you learn safety really well during the day, you have more consolidated REM sleep at night. What difference does that make? Well, here we started thinking about this idea that REM sleep clearly plays a role in the overnight memory consolidation of emotional memories. And so could it be that this increased REM sleep that we're seeing at night facilitate remembering one or more aspects of this task, which is, fundamentally an emotional experience? So to address this question, we then ran another canonical correlation where we compared the same REM sleep latent variable I just told you about to a latent variable that we called startle retention. Now, this variable was made of essentially fear recall or fear retention on day 2, safety recall, and the ability to discriminate between the threat signal and the safety signal. And so here we did see that REM sleep at night was predictive of overnight memory consolidation the next day. And as it turns out, so there's the correlation. And then as it turns out, the only recall variable that was related to REM sleep was the retention of the fear safety discrimination. So what we saw essentially is that greater REM consolidation overnight led to a stronger ability to discriminate threatening signals in the environment from safety signals in the environment. And it accounted for about 22% of the variance. So as just kind of a summary of that set of analyses, we saw that safety signal learning during the day led to greater REM consolidation at night. Greater REM consolidation at night, in turn, led to a stronger ability to discriminate threat from safety the next day. Now, interestingly, fear conditioning did not have any relationship in any of these analyses to REM sleep, which seemed very unusual to us at the time, given the animal literature. But since then, this finding has been replicated a couple of times. And so it appears that the relationship between fear and REM just might not be quite the same in humans as it is in animals. Okay, so now I want to move on and talk about the sleep deprivation aspects of this larger protocol. So here we used all three arms from the study, and so the sample size is 71. The demographics are basically the same, which makes sense because the prior set of analyses were just a subset of these people. And here we used all 3 days, right? So remember that a normal sleep group slept every night of the protocol. The pre-extinction sleep deprivation group, after fear and safety learning, underwent 36 hours of sleep deprivation. Then they had extinction learning and then sleep before recall. And the post-extinction sleep deprivation group had a normal night of sleep between fear, safety learning, and extinction learning. Then they were sleep deprived to look at the impact of sleep deprivation on extinction recall. Okay, now for the results, I'm just going to show you one slide from each of the 3 days to highlight the main findings. And on this first day, of course, everybody had been treated the same so far. So what we wanted to see was that everybody could learn fear and everybody could learn safety. And fortunately, that is what we found. So on this graph, the conditions are on the x-axis. The y-axis is the startle response. Above 0 is a threat or fear response. Below 0 is a safety or relaxation response. The black bar represents the blue circle, the threat signal. The gray bar is the yellow circle, which is the safety signal. And you can see, indeed, all 3 groups did exactly what we wanted them to do. They showed a nice fear response to blue, a safety response to yellow, and very good discrimination between the two. So far, so good. Now, on day 2, I switched the graph a little bit around here. So now what we do is we take the extinction learning protocol, which is longer than the other ones, and we break it into quarters so that we have early extinction, mid-extinction 1, mid-extinction 2, late extinction. And the bars now represent our groups. So the 2 bars that I'm showing you right now are the normal sleep group and the post-extinction deprivation group, because so far, both of these groups had sleep every night. So they've been treated the same, so they should look the same. And the early extinction block is the one that allows us to look at the overnight memory consolidation for the fear and safety signals. And here I'm only showing you the response to the blue circle, which is the threat signal. So if people successfully consolidated the fear memory overnight, they should remember that the blue means shock, and they should show a nice, robust fear response on day 2 at the very beginning, which they do. Now, as we continue to show them the blue circle with never shocking them, they should start to develop an extinction memory. And what that means is they're going to stop showing a fear response to the blue circle, which you see in this nice decreased curve here, where by the end, they're basically showing a safety response to the blue circle because now they have successfully learned extinction. So the question is, what happened to the group that was sleep deprived last night? If sleep deprivation interferes with consolidation of the fear memory, they should show a very small fear response to the blue circle today because they won't remember that it shocked them yesterday. If, on the other hand, sleep deprivation interferes with the ability to learn extinction, they should not show this nice, beautiful curve that these other 2 groups show. So what happened? The sleep deprived group looks exactly like the other 2 groups. So what this suggests is that sleep deprivation definitely did not interfere with the ability to learn extinction. And in this first block, we see that sleep deprivation did not interfere with the ability to consolidate the fear memory overnight. Okay, so now all 3 groups have successfully learned extinction. So the question is, what happens the next day when we ask them to recall the extinction? So now I flip the graph back to the way it was on the first slide, which is the conditions on the bottom, the fear response to the blue circle on the top. If sleep deprivation interferes with the ability to consolidate extinction overnight and therefore leads to impaired extinction recall, the post-extinction sleep deprivation group should actually show a big fear response, right? Because they don't, physiologically, they don't remember the extinction from yesterday. Whereas these two groups that slept last night should show a nice small fear response because they remember the extinction. Well, that's not exactly what happened. So it turns out actually the pre-extinction sleep deprivation group is the one group that showed an impaired extinction recall response. That is, they did not successfully consolidate the extinction memory overnight. So this was pretty confusing to us when we first saw this, because the sleep deprivation last night didn't seem to interfere, but sleeping last night in the pre-extinction group, they still weren't able to consolidate the memory. So then we started to realize, though, this group was sleep deprived prior to extinction, then they slept after extinction. And what that means is last night's sleep wasn't a normal night of sleep. Last night's sleep was actually recovery post-sleep deprivation. And what do we see during recovery sleep? Well, we typically see an increase in slow wave sleep. And that increase in slow wave sleep comes at the expense of the other sleep stages, including REM sleep. And so we started thinking maybe we inadvertently impaired REM sleep last night in this group that was in essence getting recovery sleep. So to look at that question, we took the two groups that slept last night, the night before extinction recall, and we calculated the REM consolidation latent variable that I talked about from the first paper. And that's on the x-axis. The y-axis is the fear response shown during extinction recall. And so what we see is that the individuals with the smallest or the lowest REM consolidation, in other words, those who actually had REM fragmentation, showed the greatest fear response the next day. That is, they showed the most impaired extinction recall the next day. So it does seem to be then that REM sleep last night was driving the extinction recall response today. And so the group that we inadvertently impaired their REM sleep also has impaired extinction today, or extinction recall. Okay, so as a summary here, total sleep deprivation did not impair the overnight memory consolidation of the fear memory. Sleep deprivation did not impair the ability to learn extinction. However, sleep deprivation did impair the subsequent sleep-related overnight consolidation of extinction, and therefore impaired the ability to recall an adequately learned extinction response. And we think this is due to the sleep architecture changes in particular, where greater REM consolidation equals better extinction recall, or in other words, REM fragmentation seems to impair the ability to recall extinction. Okay. Now, together, we think that this set of findings has some pretty interesting implications for post-traumatic stress disorder and sort of by extension, some of the fear-based anxiety disorders as well. And so remember that the clinical features of PTSD include impaired fear inhibition, both extinction and safety learning. There is an overgeneralization of avoidance, behaviorally speaking, and they chronically have fragmented sleep. So when you take these together and you use that as the context for thinking about the findings that I just presented, you come to two interesting conclusions. One, we found that REM sleep promotes the ability to discriminate safe stimuli in the environment from threatening stimuli in the environment. So if somebody has PTSD and they have chronically fragmented sleep, that's probably going to chronically fragment REM sleep, particularly if they have nightmares. And therefore, they're going to have a difficult time discriminating safety stimuli from threatening stimuli in their environment. And if you can't discriminate safe from threat, the best thing to do is just avoid the environment altogether. And then secondly, from a treatment perspective, it's interesting to note that in your gold standard exposure-based interventions, the factor that is one of the strongest predictors of success in exposure is not how much somebody habituates to an imaginal exposure in session today, but rather can they maintain that habituation across the course of the week and still show it when they come to therapy next week. What we showed is that sleep deprivation prior to extinction doesn't impair the ability to learn the extinction, which in the context of exposure therapy means doesn't impair the ability to show habituation in session. But sleep deprivation last night does impair the ability to consolidate that learning from therapy today and retain it across the course of the week. So in a patient with chronically disrupted sleep, they may look like they're doing just fine during the imaginal exposure in session, but then when they come back next week, because they haven't retained that habituation, they're going to be right back where they started this week. Okay, so I know I'm kind of running out of time. I need to end, so I'll just kind of leave it with this teaser, which is all the stuff that I just presented was in healthy controls. And while it has really interesting implications for PTSD, it really is, those implications are probably only relevant if the REM sleep fear inhibition relationship that we see in controls translates to samples with PTSD. And while I can't show you the data right now, Laura Strauss, who was a PhD student in my lab, is now an assistant professor at University of California, San Francisco, published what I believe remains the only paper in the literature looking at this relationship between REM sleep and fear inhibition in a sample diagnosed with PTSD. And what she showed is that all of the safety learning REM sleep relationships did translate into the sample with PTSD. The one thing that didn't translate was the REM sleep extinction recall finding that I showed you at the end. We don't know at this stage whether that's a sample size problem because Laura did what admittedly was a small preliminary sort of study, or whether that relationship doesn't actually exist in PTSD populations. And so that remains to be seen. So with that, I'd like to acknowledge the various sources of funding that went into this work and the related work I've done over the years, as well as a host of collaborators without whom I certainly could not have gotten any of this work done. And so with that, I'm happy to stop and answer any questions.
Thank you very much, Sean. That was lovely. Shall I take any questions, Isabel? I mean, we've got, I guess, a couple of minutes for questions if anyone wants to post them through on the Q&A.
Yeah, so I mean, I guess it's because there is like very short minutes, so the next speaker is the next speaker is already here. But I mean, you can maybe like, because I cannot see any Q&A, maybe you can. Okay, there is one, so maybe you can.
There's one about the paper by Laura that you mentioned at the end. Could we could you just say what that paper is, because someone wants to find it?
Oh, sure. It is in the neurobiology of stress, and it was published in. Let me pull my slides back up and tell you. It was published in 2018. So it's Laura Strauss, Neurobiology of Stress, published in 2018.
I have a quick question, if I may. It's in thinking back to sort of the early stages of PTSD, do you think, or sort of mechanistically, would you be thinking that sleep disruption in the early stages of PTSD is kind of embedding the symptoms there by not allowing the normal extinction and sort of safety signals to be developed?
Yeah, that's actually precisely what I think is that
right.
A good healthy sleeper exposed to a trauma, they have an acute trauma response. A large proportion of those folks will have sleep disruption. That sleep disruption then may impair the natural extinction process that allows most people to recover. And so those are the ones who may be more likely, and there's certainly data out there to suggest this, that those are the ones who are more likely to go on to develop PTSD. The other thing that we know is chronically disrupted sleep prior to exposure to the trauma. So for example, if you have chronic insomnia or you have obstructive sleep apnea, prior to being exposed to a trauma, that increases the risk of developing PTSD by 2.5 to 3-fold. And again, I think, and I have a current grant that we're about to start data collection on in the new year, looking at this question of, is the reason insomnia is such a potent risk factor for PTSD precisely because insomnia disrupts sleep in exactly the right way to impair extinction?
Super. There is a question come through, but I think in the interest of time, we should probably move on to try and keep to our schedule. So
Excellent.
thank you very much, Sean, and enjoy
you.
the rest of your evening.
Thank you.
Thank you very much, Sean. And now is the turn of Professor Tina Paunio. So it's a real pleasure for me to introduce to all of you today Tina. So Tina is one of my supervisors in Finland, working in the child sleep cohorts in 2018 together. So thank you very much, Tina, for joining us today and for finding some time in your busy schedule. So thanks a lot. I will briefly introduce Professor Tina Paunio to all of you. Tina Paunio is Professor of Psychiatry at the University of Helsinki and Research Professor at the Finnish Institute for Health and Welfare in Helsinki as well. She's also Vice Dean of Education at the Faculty of Medicine at the University of Helsinki and Deputy Editor for the Journal of Sleep Research. Professor Paunio has a special interest in genetic background for psychiatric disorders and sleep-related disturbances, and her main research focus has been on understanding the molecular mechanisms for disturbed mood and sleep and their relationship. And in today's presentation, Professor Tina Paunio will talk about some of her recent research on the genetic background for sleep-related disturbances and psychiatric problems. So Tina, whenever you are ready, so you have 20 minutes for your presentation, for your talk more or less, and then at the end there will be around 10 minutes of Q&A that Professor Matthew Broom will be leading. Thank you very much again.
Thank you, Isabel, for the very kind introduction, and thank you all, Isabel, Matthew, and Andrew, for organizing this. So it's wonderful to be here. So I will share my screen here. I'm sure it will be successful, and then I'll just put my presentation mode. Is it okay now? Can you see it? Yeah, I'm putting here. So my topic today is to discuss with you about the genetic background for sleep-related disturbances and psychiatric problems. And as Isabel mentioned, so my background is both in psychiatry and in genetics. And I became interested in sleep research some 17 years ago, and I feel that I start getting familiar with it little by little after all these years. It's a fascinating topic, and in particular, combining the knowledge on sleep medicine into that on psychiatry has been a great challenge and of great interest to me. So I have divided this presentation into 4 parts. I'm putting this just a little bit here. So first we will discuss sleep in mental disorders and about the basics, then about the genetic background of sleep traits very shortly. Third, I will present etiological models for the sleep traits and the comorbidities, and in particular, of course, from the point of view of psychiatry. And then I would ask if what are the molecular mechanisms that are involved with the phenomenon that we observe, different types of sleep traits comorbid with other psychiatric traits or with psychiatric traits. So first, basics. So sleep sleep in mental disorders in short. So symptoms of disturbed sleep or circadian rhythms are very frequent in psychiatric disorders. They occur, for example, in 90% of patients with depression and almost up to the same level in patients with anxiety disorders. But the different types of symptoms are not specific. They are not specific to any particular type of the disorder, neither are they very specific to any particular type of the patient. So that, for example, in depression, or a patient with depression can have at some point symptoms of insomnia, and then on the other period of life, symptoms of hypersomnia, just to take an example. I went systematically through the sleep diagnostic criteria for mental disorders and how the different types of sleep symptoms are presented in them. So this is one of the table in the very current actual publication by ESRS, the sleep medicine textbook. The 2nd edition just came out from press. So I think it's actually surprisingly little what we see, how the sleep symptoms are given in the criteria for psychiatric disorders. And furthermore, no systematic nomenclature is mentioned there. We talk about hypersomnia symptoms by many different words. Equally, insomnia symptoms are sometimes disturbed sleep, sometimes insomnia symptoms, and in that sense, we would need to be more systematic. Nevertheless, you see sleep-related symptoms in hypomania or mania, substance withdrawal, caffeine intoxication, depression, chronic fatigue syndrome, PTSD. You just listened to a wonderful presentation on PTSD. So there also nightmares are, of course, very, very important symptoms. And then premenstrual dystrophic disorder and chronic fatigue syndrome. In a nutshell, and this is just something I prepared myself, so take this with some precautions. But in a nutshell, we see symptoms of insomnia. This is difficulties falling asleep, frequent night awakenings, early morning awakenings, or qualitatively unsatisfactory sleep. We see symptoms of insomnia in virtually all psychiatric disorders. Hypersomnia are observed also in psychosis, bipolar disorder, in particular bipolar depression, or in depression. Unstable rhythm is also there in most of the disorders, but in particular in bipolar disorders, psychotic disorders, and substance use disorders. When we think about disturbed sleep and mental health, so we need to come to the basics of how sleep and awake are regulated. And of course, this is now clear for you that behind regulation of sleep, there is the two-process model, which determines both the circadian and homeostatic pressure, circadian system and the homeostatic pressure, and there are different brain regions that are involved there. In the end, these of course interact. But we also have mechanisms for regulation of awake, like the activating systems in the brainstem, or how thalamus is processing information and giving input into the cortical regions. So all this needs to be in balance when we talk about physiologically successful regulation of sleep and awake, which is actually needed also for good mental health in different under different environmental challenges, what we usually come across during our lives. The effects of insufficient sleep on brain are remarkable. Acute sleep deprivation leads in the end to symptoms of psychosis. After 72 hours of acute sleep deprivation, virtually all individuals experience some kind of distorted reality testing. They can have delusions or auditory hallucinations, but they come there, and this is because the systems how you test the reality of your experiences, these systems get deviated after prolonged sleep deprivation. And one of the hypotheses in my research is that people are vulnerable in different amounts for how they react to sleep deprivation. There are individuals who tolerate better sleep deprivation than the others. This is something we already know beforehand. Chronic sleep insufficiency is a different phenomenon. There, people can sleep, or they have the opportunity to sleep, but for a reason or the other, they don't sleep enough. Usually, the reason behind this is related to insomnia and to the fact that they have either difficulties in falling asleep or difficulties maintaining the sleep. And from epidemiological studies, we know very well that insomnia increases risk for a number of disorders. The most remarkable risk is for psychiatric disorders, and there in particular for depression and anxiety disorders. But you also see increased risk for type 2 diabetes, cardiovascular diseases, dementias, in particular Alzheimer's diseases, and according to some studies, also to psychotic symptoms and alcohol abuse. Here I would like to mention that for anxiety disorders, it looks like it is in particular the vulnerability to have problems in falling asleep that is typical for anxiety disorders, while for depression, the picture is a little bit more complicated. Okay, then let's see, we move on. Then about circadian disruption and mental health. So misalignment of an environmental signal with the endogenous circadian rhythm, this is something we call for circadian disruption. And very frequent reason for this is if somebody is working irregular hours or well, traveling across time zones. This has not been so prevalent during past months, but perhaps it becomes more frequent again after we have gotten over this pandemic era. Then in our studies with Sonia Sulkava, we focused finding on individual liability to circadian disruption. And in this particular study, which I present here very in summary, the results for Sonia first performed genome-wide association study on a very small number of individuals, only about 200 individuals, for their symptoms of fatigue if they worked in shift. So shift burnout in relation to shift work. And there she studied more than 1 million variations after all imputation, the methods you use for genome-wide association studies. And from all of these variations, the only potentially significant finding was for a variation that was located close to melatonin receptor 1A. This finding was replicated in a few more samples so that we then reach the genome-wide significance level, just, I must say, but nevertheless. So I think it's quite a coincidence that we observe a variation at melatonin receptor 1A that increases the risk to experience burnout if you work in shift. And then in preliminary analysis, Sonia was able to observe that there might be epigenetic mechanisms that are involved in this liability, so that if you have this specific risk variation, which is quite frequent in the population, and if you work in shift, then the DNA methylation pattern at promoter for the melatonin receptor 1A is different. But this is something we need to study further. So this finding is only preliminary. Okay, then we continue. Then about genetic background of sleep traits. So very quickly about this. So heritability estimate for self-reported sleep traits are only modest. It typically is something between 20 to 45%. An exception here is chronotype, where the heritability is around 50% or 55%. In this particular study on right, we were looking at the relationship of the genetic or the heritability for poor sleep quality and depressed mood, and found that their correlation was relatively weak, up to between 20 and 40%. But what in this modeling we then found was that if you had the risk for experiencing poor sleep, as here in women, and something happened in between, which we don't know exactly, then, but some environmental triggers happen, but then you had, after 5 years, highly increased risk for experiencing a depressed mood. So compared to many other traits, the heritabilities for sleep, subjective sleep traits are at most high. And the same thing was observed also based on the UK biobank data here from a few years ago, but the findings are still of the same relative levels. So that, as for example, for standing height, you have in heritability up to 70 to 80%. So for insomnia, it's around 10%, or morning evening type person below 10%. So this is SNP-based heritability we are talking here about. So this is something to bear in mind. So what this indicates is that even though our sleep and biological rhythms, in the end, they are determined by the genome, so environment modulates them heavily. And this is also meaningful. Sleep needs to be modulated by the environment. And that's why we don't see that high heritability estimates in populations, because people live in different types of environments. Nevertheless, the large biobank studies have been successful in finding genetic influences on sleep traits. And here are a few examples of the studies of very high quality. And according to these studies, here I took an example by Janssen et al. A couple of years ago. They looked then the expression patterns of the genes that were identified in these studies and how these RNAs are distributed in the different brain regions. Nevertheless, what is most important perhaps from these studies, that based on that data, you can calculate polygenic risk scores, which then gives you the intrinsic quantitative amount of your liability to experience insomnia or evenings type of chronotype or short or long sleep duration or sleepiness or other variables. We looked with Samuel Jones and Hanna Olela how the different sleep PRSs are correlated, and found, this is also found in the original publication, but we combined all this data. So we found that the genetic risk for insomnia and short sleep are very highly correlated. So their correlation coefficient is more than 60%. And then that we can see some correlation between the risks for insomnia and fragmented sleep and short sleep and diurnal preference or morningness. To me, these correlations are surprisingly low. The only exception here is the high correlation between insomnia and short sleep. So it appears that there are different genetic variables that determine the liability for these subjective sleep traits that have been studied in the UK Biobank research. How then is it possible that we see so much comorbidities between sleep traits and their comorbidities? So you can have three different possibilities. First, disturbed sleep causes mental disorder. Second, mental dis causes disturbed sleep. Third, you have shared risk factors for disturbed sleep and mental disorders. And evidence for all these possibilities exist. However, if we look at depression and sleep, insomnia, so at least our studies show that it is clear that insomnia precedes depressive disorder and not vice versa. But for anxiety disorders, the pattern is more complicated. On the other hand, from these UK biobank studies, as I mentioned earlier, one has been able to observe very important shared risk factors with high correlation coefficients over 50% between, for example, anxiety disorders and insomnia. Then if we look at the systematically of these correlations, how the psychiatric polygenic risks are correlated with those for subjective sleep traits. So this is something we have been working again with Samuel and Hanna. So we found moderate or high level of correlation between the genetic risk for insomnia or short sleep. Remember they correlated with each other, and depression, anxiety, PTSD, ADHD, and smoking. But also there, the correlation coefficients, they are high, but they are not extremely high, I would say, not more than 50% in our analysis. We observed also some correlation between the risks for evening type of preference and schizophrenia, autism spectrum disorders, and alcohol consumption. What we are for the time being doing is looking at by using Mendelian randomization about the potential causalities between these correlations. So in the end, I would like to ask if there are epigenetic mechanisms that are involved behind these high level of comorbidities we observed in clinical work. And this is something we have been looking also, again, quite systematically. This particular study has been performed in collaboration with Deborah Skeen and her team, where we found that in acute sleep loss, where the DNA samples were collected every 3 hours, we see accumulation of DNA, both hypomethylation and hypermethylation, but in particular hypomethylation, I mean epigenome-wide. So we observed changes in blood leukocytes DNA methylation after prolonged sleep deprivation, but the sample is small enough at this point to say which particular genes are for sure affected here. But the pattern is clear, and it's arising after 27 hours of DNA of sleep deprivation. So this would give evidence that the epigenetic processes are involved if the sleep is disrupted. In another study, which was published 2 years ago by Alexander Lahtinen, we found that in chronic sleep loss, we see also hypomethylation of DNA globally in epigenome, and so that 78%, so 4 by 5 of the differentially methylated positions were hypomethylated in cases who suffered from insufficient sleep, as compared to those who did not suffer from insufficient sleep. We also observed clustering of signals on genomic loci that had been previously linked in visual processing and circadian regulation, so giving some biological evidence that our findings are not only false positives. We have to always, of course, be aware of that possibility because we do perform so many tests. And then the pathway analysis evidence for compromised processes of neuronal plasticity. So very general findings, I would say. In another study, I would not go in detail here, but this was done in adolescent boys who suffered from insomnia and depression as compared to healthy controls, and those cases have problems in their slow wave sleep dissipation. So we observed some particular sites in genome that were differently methylated, and then pathway analysis gave evidence that there would be synaptic long-term depression pathway which could be involved, which could then link these problems in sleep and mood for perhaps for memory processes. And one of the sites also linked to the flattened pattern of slow wave sleep observed in the cases, but it was also quantitatively observed in controls. So here is the data for that particular site. So some evidence for the biological mechanisms behind our observations. In the end, I just show you our latest report by Alexander Lahtinen, where we found that these changes which we observed in shift work, changes in DNA methylation, so at least some of them are changeable, so that they disappear. The differences between cases and controls appear after a period of vacation. And in a general pattern here again was that we observed hypomethylation among those who suffered from shift work disorder. And then there was some implication of the specific pathways related to NMDA glutamate receptors that were affiliated if you suffered from shift work disorder. So I would like to conclude here that symptoms of disturbed sleep or circadian rhythms are very frequent in psychiatric patients and disorders. And despite any lack of specificity, there are some patterns that arise from epidemiological and genetic studies. We need large data sets for that, in particular in the genetic studies, but we have them from UK Biobank. The comorbid pattern of insomnia gives evidence for the shared etiological background and secondary effects of sleep insufficiency on brain and emotions. But the pattern for diurnal preference, according to our and other studies, is much less clear. But there is some evidence for the effect of the compromised mental health leading towards evening type of preference. So I would like to end at this point. I'm happy to answer to your questions if you have any. So thank you.
Thank you very much, Professor Paunio, for a great talk. I've covered a lot of data. That was really fantastic to hear. So yeah, we've got about 5 or 6 minutes for any questions from colleagues who attend the webinar. So please pop your questions in the Q&A for Professor Paunio to answer. One thing I'll quickly ask when people are typing in their questions, if that's okay, is a kind of treatment question, if that's right, Professor Paunio.
Yeah.
you helpfully outline the various kind of hypotheses people might want to test between mental disorder and sleep problems, how this is connected. Does that translate to how one should think about therapeutic interventions, whether one should treat the sleep disorder primarily or treat the mental health problem primarily? And I'm aware you said the story with depression and anxiety might be different. So I guess that'd be one question I'd like to ask you about how that should impact on treatment choices. And secondly, how far is the epigenetic story away from any translation to practice?
Yeah. So about the treatments, I think it's absolutely important that in psychiatric patients that we treat the patients and not the disorders. So it's very good that we have these different protocols for treating like schizophrenia or depression or whatever. But always when you have the patient, a patient is an individual, and you need to, as a doctor, you need to be aware of what is the specific pattern of symptoms within that particular patient in relation to his or her life history also. And there's lots of data evidencing that if there are prominent symptoms for sleep, such as for insomnia or a disturbed circadian rhythm, you need to take care of specifically of that if you treat the patient. What is usually happening is that the patient comes with depression, you start the treatment of the depression and you follow up, and then you also, oh, there's also insomnia, perhaps I would like to treat that too. I think that the doctors should take that into their consideration already during their first meeting with the patient. But here, I'm not perhaps a typical psychiatrist in that sense, so that I'm just giving my view. Then about that, if we could apply DNA methylation studies in clinical work. So actually what we are right now doing is that are there any diurnal variation in the DNA methylation patterns? I think there might be. And I think that it's a very complicated story, and making their clear clinical applications, it's not like decades for it, but we need more data for that. But I think it might be one alternative for biomarkers for disturbed sleep. But we need also other biomarkers, and of course, it would be nice to have more brain-specific biomarkers. Here we have only been able to analyze blood leukocytes, but you could also, of course, do skin biopsies or saliva, but taking brain biopsies is not an option.
Thank you. And you mentioned, I think on your maybe penultimate slide, about the connection with long-term depression in adolescent boys. It just intrigued me. Was that a potential mechanism linking sleep disorders with memory impairment or cognitive impairment, perhaps? I just wondered how solid that connection was with kind of behavioral neuropsychological findings.
Yeah. In that particular sample, yes. So there we observed the varying DNA methylation pattern between the cases and the controls, and some of these sites are related to the problems, objective problems in sleep. We did not observe a relationship between these DNA methylation positions and cognitive measures. And how we interpreted this is that they were still young boys who were perhaps able, or who were likely to be able to compensate their situation. And what I would have very much like to do is to follow them further and see how they develop. And as we know also in depression is that if you have chronic depression, then you start getting also permanent problems in your cognition. If you have acute depression, usually you recover, but then getting chronic, you get perhaps permanent deviations in cognitive processes. And that's, by the way, one of the reasons why we need to treat depression. It's not a disorder to just follow up, but we need to treat it.
Fascinating, thank you. I was making just a brief comment. I mean, I guess the data on shift workers was also fascinating. Yeah. And one of our colleagues in psychology here at Birmingham is working on sleep deprivation in parents and the effects on social cognition. So it's a topic that's very close to many of us with young children. So thank you very much for your talk today, Professor Paunio, and I'll hand back to Isabel, I think, for the next session. That was fantastic. Thank you.
Thank you.
Thank you very much, Tina. And now is the turn of Professor Timo Partonen. Timo, I think. Yeah, you're here.
Yes, I'm here.
So thank you very much, Timo, for joining us today. And so it's my pleasure as well to introduce Timo Partonen to all of you. So Timo was my supervisor at the Finnish Institute for Health and Welfare from 2016 to 2018, but we still keep in touch. So thank you very much, Timo, for accepting this invitation. I will briefly introduce to everyone. Professor Timo Partonen is research professor at the Finnish Institute for Health and Welfare in Helsinki. Professor Partonen was one of the pioneers to investigate seasonal affective disorder, and his work was published in The Lancet in 1998. His key interest is to study the effects of environmental factors, health-related behaviors, and endogenous factors on the phenotype of mood. And today, Professor Partonen will talk about the relationship between chronotype and mental health, and how night owls tend to have more mental health and health-related problems than early birds, such as mood disorders, anxiety disorders, substance abuse, insomnia, or sleep apnea. So I think like this talk as well, I think ends very well, like especially I think it was in the relation to the first of the talk with sleep ADHD, that as we're talking about the circadian rhythm as well. Now Tina, she was talking as well about some genetic background for chronotype. So thank you very much, Timo. You have around 20 minutes for your presentation, and then we will end with 10 minutes for the Q&A. So thank you very much.
Okay. Yes, so thank you for the invitation, Isabella and others who have organized this webinar. So the topic is chronotype and mental health. And assessing the chronotype you can assess it by measurements, so-called objective measurements. Example, using accelerometers to record the diurnal cycle, but those cycles are rest activity movements. Whereas if you measure rectal temperature using rectal sensors to record, you can have the circadian rhythm of core body temperature and assess the chronotype by that mean. Or using ingestible sensors as well, or dermal sensors to measure distal skin temperature. So by that way, you have continuous records of temperature, and you can track the diurnal cycle, or in fact the circadian rhythm during the day and during the night of core body temperature or distal skin temperature. But however, for epidemiological studies, those are very hard ways of measurement. So usually we use self-reports. So you can assess the chronotype by using self-reports. Maybe one of the most widely used is the morningness eveningness questionnaire, MEQ. It was published in 1976 by Horn and Östberg, and it determines the preference for timing the daily activities. And it produces a score, a sum score, a total score. It has 19 items. I'll show you here just the last item of the morningness eveningness questionnaire. It's the item 19, and it asks whether one hears about morning and evening types of people, and which one of these types do you consider yourself to be. And there are 4 options to answer: definitely a morning type, then rather more a morning than an evening type, or rather more an evening than a morning type, and then definitely an evening type. And for example, UK Biobank study uses only this one item from the original morningness eveningness questionnaire to assess the chronotype. Then there's another widely used self-report. It's Munich Chronotype Questionnaire, MCTQ. It was published in 2003, and it aims to determine the phase of entrainment by assessing the middle point of sleep, and produces a clock time. So the basics is different as compared with the morningness eveningness questionnaire. So this Munich Chronotype Questionnaire asks only about sleep behaviors on work days and on free days. So when do you go to bed? When you decide to fall asleep, how long it took to fall asleep, and then when you wake up without or with alarm clock, and when did you actually get up. after waking up. So the target is to assess the average midpoint of sleep on 3 days, and it equals to chronotype according to this questionnaire. But of course, there's also average midpoint of sleep on work days that is usually different. And this difference creates the so-called circadian misalignment. So misalignment between the local time, clock time, and the internal clock time. So external clock time and internal clock time are not aligned with each other, but there's this misalignment. And if this continues longer time, it may create circadian disruption. And what is this internal time or circadian time? It's we have in our body circadian clocks. They generate and maintain rhythm, so-called circadian rhythms, and also other rhythms of diverse periods. But there's a master clock. The principal clock is located in the brain. It's in the hypothalamus, in the anterior part of the hypothalamus. It's a pacemaker, and it conducts time signals to the body, the rest of the body. It's like a conductor of a symphony orchestra, and it targets to create circadian resonance. So all these peripheral clocks have their own time, own internal time, but this master principal circadian clock or pacemaker try to create synchronization between all these clocks in our body. So there's genetic factors which influence on how this circadian clock system works, and also environmental factors influence how this circadian clock system works. If these are aligned with each other, then there's no desynchronization, but there might be this circadian disruption, and it may create problems in the longer term. So there are evidence for cardiovascular diseases, inflammation, diabetes, type 2 diabetes, obesity, and mental health disorders that are at least partly due to circadian disruption or circadian misalignment. And chronotype is it's a behavioral trait. It's a phenotype that is under these genetic factors and environmental factors. So this chronotype refers to the preferred timing of behaviors, not only sleep, but also other activities that we take during the day and night. And this behavioral trait is under the influence of genetic factors and environmental factors, half and half about. There are estimates using twin studies that the overall genetic effect, so-called broad sense heritability, is about 50%. And the remainder is accounted for by environmental factors that are not shared by twins. And what is the stability of this behavioral trait, chronotype? We have now collected data from 2 population-based studies from Finland. It creates the design of a longitudinal study for nearly 3,000 individuals. And it's over 7 years. These are middle-aged participants, and there was no change in chronotype in 70% of them. Then there was a change towards more eveningness in 16% and towards more morningness in 13 or 14%. But these shifts from one extreme to another are rare, either from night owl to become a morning lark, only 0.5%, and vice versa to the similar extent. Then we have another longitudinal study that spans over 20 years now for 567 persons. They were former top athletes of Finland, and they represented Finland in World Championships and Olympics during the 1920s, 30s, 40s, 50s, and 60s. And they were assessed for their chronotype for the first time in 1985 and the second time in 2008. And we saw that there was no change in chronotype in 55% of them. There was a shift towards more eveningness in 20% and towards more morningness in 26%. But here as well, the shifts from one extreme to another were rare. So it seems that this master circadian clock in the brain and the timing of night sleep, they contribute to health status. There's this study of Australia. It's on school children aged 9 to 16 years, over 2,000 of them. It created a design that then assessed if sleep of equal duration was present, but the sleep was late in its bedtime as well as in its wake-up call. So this created the increased risk of weight gain, obesity, sedentary physical activity, long screen time, and unhealthy diet in children. And we have collected data from school children aged 14 to 20 years from Finland. And it's a population level study. And we found that the individuals with greater eveningness had a heightened odds for tiredness, trouble falling asleep, and nightly awakenings, headaches, pains, irritation, anxiety, depressive symptoms, as well as troubles with school performance, with 2 exceptions. So there was no troubles with teamwork performance, nor getting along with school friends. Then we have analyzed these population-based studies. I'll show in the end what these are, these databases. But all these are at population level data sets. The individuals with greater eveningness have more frequently, this concerns now adults, more frequently insufficient sleep, insomnia, nightmares, and the use of sleeping pills for these sleeping problems. Among pregnant women, especially, the individuals with greater eveningness had more frequently insomnia and troubles in falling asleep, poor sleep quality, as well as among their offspring, late bedtime at the age of 3 and 8 months, and prolonged sleep onset latency at the age of 18 and 24 months. Then we know that from our databases that at population level, the individual with greater eveningness had more frequently emotional eating, and they had greater weight gain, an increase in body mass index over 7 years, although only among women, and unhealthy dietary intake. And some details of the diet that was unhealthy are shown here as well. And on the weekend, this pattern was more pronounced, as well as eating occasions were more frequent, but meals were more irregular. And then this physical activity, not only sleep and diet, but also physical activity, seems that at population level, the individuals with greater eveningness had more frequently very low physical activity or none, and more sedentary behaviors, more sitting time, more time spent sitting. Then, of course, all these might have influence on mood as well. But then to look at specifically mood, we found that at population level, the individuals with greater eveningness had heightened odds for mood and anxiety, not only on symptom or disorder level, but also on hospital admission level, indicating this, the severity of these symptoms and disorders. An example concerning depression, depressed mood, anhedonia, depressive symptoms, a diagnosis by medical doctor or treatment received for depressive disorder, including antidepressant medication, all these odds ratios were heightened. Then there's a specific type of depressive disorder. It's called seasonal affective disorder. It means recurrent depressive disorder with seasonal pattern. Usually it is so that during wintertime, there's these depressive episodes that keep repeating themselves, reoccurring winter after winter. And we found that SAD, seasonal affective disorder, was more frequent among night owls compared with intermediate types or morning larks. And we can look at the other way around, that among SAD individuals, the most of them are night owls, and then followed by intermediate types and morning larks. So it seems that health hazards accumulate on night owls. And we have now found that night owls have more frequently insomnia, nightmares, depression, winter blues, hypertension, type 2 diabetes, bronchial asthma, spinal diseases, back pain, as well as sleep debt, unhealthy diet, emotional eating, smoking, high alcohol use, low physical activity, long screen time, for example. And what is the cause and consequence? So this kind of a relationship. Is it so that the evening is the primary issue and health hazards are the secondary issues? It seems so, and not the other way around. So eveningness, there are some longitudinal studies that has assessed this eveningness and different kind of health hazards in the same individuals over time. And the follow-up time might be 1 up to 3 years. And it seems that eveningness predicts more frequent depressive symptoms as well as more frequent depressive disorders. And it seems also that eveningness predicts worse academic performance, more substance use, greater social jet lag over time during the follow-up. Whereas substance use predicts greater social jet lag, but not eveningness. Those were the Northern American studies. And then there's this elegant study, what can be done? So does eveningness create health hazard? Not necessarily, because a phase advance strategy had a positive impact in this English study. So it was a study of evening types only that were divided into 2 groups. And the other group received 1 advice only, and it's involved here on the left column. So participants were asked to eat lunch at the same time every day, whereas the other group received all these instructions and advice on the left column. And the positive impact was clear, because the feelings of distress were relieved within 3 weeks. Depressive symptoms, although mild, were alleviated, and physical performance measured as with the hand grip measurement was improved, for example. And of course, these individuals slept better. Why this happened? Usually, when you try to advance your circadian rhythms or diurnal rhythm, you receive light through the eyes in a different time of the day. And more light you receive, more light you are exposed to during morning hours, it advances circadian rhythms inside our body. And evening types, often they have a longer internal day, and usually they have phase delays that are greater than other persons. And usually it's beneficial to advance the rhythms. And light exposure is very effective in advancing circadian rhythms if the exposure is during the morning hours. Whereas if light exposure is during evening hours, it might be harmful. And then you need to also adjust your sleep-wake schedules, meal rhythm, and a timing of physical activity to have the optimal outcome. Because your feedback causes advances or delays in circadian rhythms, and it can increase or decrease circadian misalignment there might be in your rhythms. So a behavioral change can speed up or slow down the master clock. And here is example, the timing of light exposure, whether it creates delays or advances, melatonin pills, whether they create advances or delays, and physical exercise, whether it creates advances. It's shown here in the graph. And this is a serious issue because it seems that chronotype is associated with premature mortality. This is our finding from the former top athletes study, showing that those who were evening types died younger. So it became evident after the age of 56. And it's agrees with the other findings. So University of Manchester longitudinal study of cognition in normal healthy old age cohort, and UK Biobank study as well. So in the end, these are the databases I was referring to. So we have these population-based surveys for health examination studies, Health 2000 and Health 2011. It creates a longitudinal design, FinRisk 2007, and DILCOM 2014. It creates a longitudinal design, FinRisk 2012 and FinHealth 2017. And all these included a random sample of 10,000 adults of the general population, adult population, and their collections were transferred to TAS Biobank. And then we have nationwide school health promotion studies as well. And here are my acknowledgements to my co-workers from THL, University of Helsinki, Tampere University, Karolinska Institutet, and Medical University of Vienna. So that's all this time.
Thank you very much, Timo. So I think like we all like have learned, so I think it was like a really clear presentation, and especially now for me, it's a bit, I had a question about the treatment, for example, but now I think it was like really clear in my mind. I had something to ask you, but first I will start with Andy, that I see that he has raised his hand. Yeah, you go first.
You can go first if you want.
No worries, go ahead.
Thank you, Timo. That was really interesting. I just wondered the negative impacts of eveningness. can you disambiguate the impact of sleep debt and misalignment from that? So, I mean, I guess the other option would be that society allows people to be an evening type if they want to be an evening type, and you know, there's less pressure to fit into a misaligned type. So I guess that's the question. You know, do you think it's best to try to shift evening types earlier to reduce the negative impacts, or should you allow society to become more flexible so that there's less sleep debt and there's less misalignment?
It's a good question. Of course, you can avoid sleep debt by many means, I think. We have, let's say, control for the sleep depth in some of our analyses, and it doesn't take away all the negative effect that eveningness has on these outcomes we are interested in. So sleep depth itself doesn't explain all these negative health effects that are associated with eveningness. But I think your study, where you were involved in that English study I referred to, showed very nicely that you can adjust your internal time and have beneficial effects by advancing. So also for evening types can, and I think that they could at least think about advancing their timetables earlier, because it seems somehow that because evening types have longer internal day, so longer period of circadian rhythms, I think the key is there. It somehow creates a negative, let's say, situation, kind of a disorder inside the body that can become harmful in the longer term.
Thank you, Timo. So now we have a question from the audience. So this is from Siam, and he says, excellent talk. You said that genetic and environmental factors have about equal influence on chronotypes. However, which of those two are more important in circadian disorders?
Circadian disorders. So circadian sleep-wake disorders, for example. Well, it's half and half, I think, and it's a positive side of this because the effect of environmental factors is about half on this phenotype, chronotype. It means that you can really influence the situation. So there are environmental ways of getting benefit. So phase advancing strategies, for example, or in some cases, you may want to delay your circadian rhythms, example, after transmeridian flights, for example. And then you can take these measures that change the length of the internal day, so light exposure, for example, into use. So right-timed light exposure can advance or delay your phase. And it depends what the situation is. Also for shift workers, it may help. If you create a schedule for light exposures, right-timed light exposures, you can alleviate many of the symptoms that shift workers usually have because of the shifts.
I have a question, like, so in the beginning of your presentation, you were explaining the difference of the mornings and evenings questionnaire and the Munica questionnaire. So which of those ones, because what I've seen in the literature as well, we were working together in this, right? So then I've seen like, so independent, I guess, so some people might prefer one question or the other. Which would be like your recommendation? Of course, I guess it's preference, might be subjective, but anyways, which would be your preference in between these questionnaires? So which one, I guess, in terms of defining a person that is evening nurse, for example, so which would be more accurate in that sense?
No, they track the same phenomenon, of course, but morningness, eveningness questionnaire asks more questions, not just sleep-related questions. So it asks also about other activities than sleep. Munich chronotype questionnaire is it focuses on sleep behavior only. But if there's a possibility, use both. There's also ultra short version of Munich chronotype questionnaire. So only 2 questions. And then you can take just the last question of the morningness, eveningness questionnaire. It gives about 83% of the all information that 19 items give you. We have made this kind of a psychometric analysis, but of course it's not 100% then, but we have used 6-item shortened version of morningness, eveningness questionnaire. So under 10 questions, you may have really good overview of chronotype for epidemiological studies as well.
Yeah, because I guess it's like one of the things. So I guess sometimes when people might think about chronotype, or me at the beginning before I started working with you, maybe so I was thinking of more about sleep, but then as well this is, and I think it was very clear in the treatment, for example, because you might say, okay, like let's just go earlier to bed, but some people might say, yeah, but I cannot fall asleep. So what I understand from your presentation is all the different patterns should be as well like advanced. So physical activity, eating earlier. So I guess all the other activities. So finally, everything is interconnected, right? So then in that sense, I was thinking the morning and evening is covering maybe a bit, wider all the aspects that and mornings, evening will refer.
Yes, and usually for behavioral change, you need to think about light exposure, sleep, meals, and of course diet, what you eat, but especially when you eat, and physical activity, the timing of these daily activities.
And then I guess that as well that later affect as well like the sleeping times in that sense, or like maybe you feel more sleepy if you do everything earlier. And
yes.
Yeah. So I don't know if there is so I don't see any more questions from the audience. So then I think this is the end of your session and the end of this webinar. So thank you very much again, Timo, for coming today, or participating today. And thank you very much to all the speakers. Thank you as well, Andy and Matthew, for co-hosting today this webinar. And thank you to all of you who attended today. And there is the next week, there is the next session of the webinar on Wednesday from 1:30 to 3:30 in UK. So I hope to see you all there.
Yeah, thank you, Isabel, and all the speakers. Have a great morning. Yeah.
Thank you, Matthew. Okay, so then I will end the session. Goodbye.